Thank you, Marilyn, and a very, very special welcome to all the Burnet supporters here today. A special welcome to the Burnet ambassadors, members of the Burnet and the Alfred and Monash, members of laboratories, particularly my own laboratory. Thanks very much for all coming today. A very special thank you also to our two speakers, John and Deanna. Took incredible courage to tell their stories, and what an amazing inspiration to hear how people such as Deanna and John have lived with HIV and used it to really empower their lives. And I hope what you also have learnt from them is what incredibly productive lives they lead, and how very different having HIV in 2013 is to having HIV 30 years ago. And at my own table I've met a number of Burnet supporters who lost their sons in the early 90s, and how very different things were back then. So what I hope to convey to you is the incredible contribution that science and research has made for people living with HIV, and talk about what the future might bring if we could maintain that level of success that science has had in the last 30 years. So I work on what was once a controversial area, because no one wanted to talk about a cure for HIV. They called it the C word, because no one was too scared to talk that we could contemplate a cure for HIV. But I hope what I can share with you today is how much the field has changed, and how differently many people feel, including many scientists, about the possibility of one day finding a cure. So antiviral therapy, or antiretroviral therapy, is probably one of the greatest medical success stories of the last century. And I say that as a practicing physician. I'm experienced in many areas of medicine. It truly is an amazing success story. This is a picture on the left of what anti-HIV therapy looked like in 1996, and what it looks like now on the right, where literally most people with HIV can effectively control their virus with one tablet a day. The one tablet a day contains 3 different types of anti-HIV medicines, but it's and has a huge impact on people's lives. It's literally changed a universal death sentence to a lifelong, chronic, manageable disease. And for many people who start treatment at the right time, life expectancy is now normal for people living with HIV. And it's incredible to think we've achieved that in just 30 years. Added to that, HIV drugs make a huge effect on people's infectiousness. So a person who takes HIV, who takes anti-HIV drugs, their infectiousness is reduced by 96%. So the drugs also have a very big impact on the spread of virus within communities. And added to the great success of finding these drugs and having them available to people is having these drugs available in low-income countries. So this is a graph showing the number of people taking antiretroviral therapy, or the acronym we use of ART, or antiretroviral therapy, in low and middle-income countries across the world. And you can see back in 2003, virtually no one in these countries had access to this life-saving medication. In 2012, close to 10 million people, or 65% of people who need treatment and on treatment. And back in the early 2000s, most people thought this was completely impossible. They thought treatment was too complicated, it was too expensive, we would never be able to deliver it. So this is an amazing illustration of the combination of great science to get the drugs, but then incredible advocacy and impact on policy, impact on pharmaceutical company regulations to get the drugs into low-income countries. And the projected number of people that will one day need anti-HIV therapy by 2015 being 15 million people.
Sorry,
yes, we have a question.
I can't see from here what the colours mean.
The colours illustrate the regions in which the people are receiving these drugs. So blue is Africa, green South America, yellow Southeast Asia. The total value is 10 million, most of whom are living in Africa, which is the main site of the epidemic. So if treatment is so good, why do we need to even think about a cure for HIV? And I think there are three pressing reasons why we need a cure for HIV. And the first is that although life expectancy is normal in people who start treatment early, it remains reduced in the majority of people who initiate treatment once they have damage to their immune system. And we also know there's ongoing morbidity or ongoing illnesses in people who take treatment, partly from the side effects of the drug, but also partly because the virus is always there at very low levels. Amazingly, even despite these incredible success of getting antiretroviral therapy into low-income countries, for every 2 new patients who initiate antiretroviral therapy, there are 5 new infections. So the number of infections continue to occur. And finally, and I think this is really one of the major reasons, is that funding lifelong antiretroviral therapy for all who need it may not be sustainable. And I don't for a minute mean that this is relevant for countries such as Australia or the US or Europe, but in low-income countries this comes at its very significant cost. So just to give you a sense of that, the cost to maintain 6 million people back in 2010 on antiviral therapy was around $15 billion per year. And the estimated cost to get 15 million people, who is the number of people who currently need treatment, is estimated to be $23 billion. So that's a number that's a little hard to digest, but just to put it into perspective, AIDS treatment costs alone of the $23 billion would account for half the US foreign aid budget by 2016 and all of it by 2024. So the costs, particularly of delivering antiretroviral therapy in low-income countries, are very significant. So if we could find a way that people could take treatment for maybe 5 or 7 years, but not 40, 50 or 60 years, this could make an enormous difference to the number of people who can access treatment. So I hope I've convinced you that we do indeed need a cure for HIV. The next question is whether a cure is even possible. And first of all, what do we mean by a cure? It means different things to different people. Traditionally, in infectious diseases, a cure means getting rid of the infection completely. When you go to the doctor with tonsillitis or a skin infection, we mean eliminating the infection completely. So to do this in the setting of HIV, we would have to eliminate all HIV-infected cells. We would have to be sure that the virus could not be detected in blood, and this is often or now referred to as a sterilising cure. And I think most people believe this is actually going to be an incredibly difficult thing to achieve, and I will explain to you why. What might be more realistic is to think of HIV like cancer, or think of a way we can put the virus into remission so that the person would have long-term health in the absence of anti-HIV drugs. They would have virus still there, but it would be at very low levels and not causing damage to the immune system. And this is now commonly referred to as a functional cure. And there's a lot of evidence now suggesting that a functional cure may indeed be more possible to achieve. So is HIV cure possible? Well, over the last 4 years, we've heard of many examples where HIV cure is possible. They're rare and they're all in unusual settings, but we've learnt it is possible. And the first report of cure was of Timothy Brown, who was initially called the Berlin patient, but he too had the courage after several years to come out and put his face to this amazing story. And Timothy Brown is an American man living in Berlin at the time. He had HIV and was on anti-HIV medications and developed leukemia. And he needed a bone marrow transplant to treat his leukemia. And the doctor looking after him at the time had a brilliant idea to use a bone marrow that came from a person who is naturally resistant to HIV. So in this room, about 1% of you are naturally resistant to HIV. And the 1% are resistant to HIV because you don't have a particular molecule that the virus needs to enter your bodies. And that molecule, and I'm going to mention it because it's quite relevant to some of the exciting work being done now for cure, is called CCR5. So about 1% of Caucasians are CCR5 negative, and they're resistant to getting HIV. So the donor for Timothy Brown was a CCR5 negative donor. So the Timothy Brown received the donor's bone marrow, and then shortly after the transplant stopped anti-HIV drugs, and now 6 years after the transplant, there is no virus detectable anywhere in Timothy Brown's body, not in his blood, not in his gut, not in his brain. There is no virus detectable. He has a sterilising cure. He's the only example of sterilising cure. Now, we don't for a minute think that transplantation is the answer to curing HIV, but it tells us it's possible. And now for many of us, the idea is to take that story and try and work out, what was it? Do you need a transplant, or was it one part of his treatment that we could mimic in some other way? Now, since that time, there's been 2 further reports of cure after transplantation. These were the Boston patients who were only reported just 2 months ago. And these were 2 men, also with cancer and HIV, who received a bone marrow transplant. But in these 2 cases, the donor was not CCR5 negative. It was a normal bone marrow. And these patients too have now stopped their antiviral drugs, only about 2 and 4 months ago now, and the virus hasn't come back. So we're just beginning to try and understand what was it about the Boston patients that led to their currently what we would call a functional cure. And finally, the Mississippi baby, the story of a young infant born to an HIV-positive mother who received anti-HIV drugs within 30 hours of birth, who now is 2 years old and been off treatment for 6 months. She too is cured from HIV. And another group of patients in France, the Visconti patients, who also started treatment early. 14 patients who stopped their drugs and the virus hasn't come back, present at low levels. So these stories, or anecdotes, what we call them in medicine, are not a cure for HIV. Giving very early treatment within 30 hours or a bone marrow transplant could never be introduced and be of benefit to the 30 million of people who live with HIV. But they tell us it's possible, and they give us a whole lot of new ideas to pursue in the laboratory of how we could try and mimic the descriptions of these patients. So just very briefly, why can't current drugs cure HIV? What's so mysterious about the virus that it won't go away like other viruses and bacteria that many of you are familiar with? Well, this is the only graph I'm going to show, so don't panic. It's not going to get a whole lot more scientific. But this is just an illustration of what happens to the HIV in blood, shown in red, and someone's immune system, shown in blue, when they go on treatment. And when people go on treatment, the amount of virus in the blood rapidly declines to what we call undetectable levels. In the clinic, we can measure virus down to around 50 copies per mil. And within a month, pretty much everyone that takes anti-HIV drugs can have an undetectable viral load. And at the same time, in most people, the immune system will slowly recover. It takes a bit longer, it's a bit more variable, but in most people it will reach normal levels within several years of treatment. But in pretty much everyone, except the few cases I talked about earlier, the Mississippi baby and Visconti patients, if you stop anti-HIV drugs, within about 2 to 3 weeks, in pretty much everyone, the virus rapidly returns and returns back to where it started. So one area that many of us are working on in the Burnet is understanding where the virus is hiding when the viral load is undetectable. And where does it come from when it rebounds after stopping treatment? And we actually know a lot about that now. And we know that the virus, the main place where the virus hides, is inside the patient's DNA. So this is just a representation of a cell in grey, a person's DNA in white, and the virus buried inside that person's DNA. And HIV has the capacity to basically get inside a person's DNA and effectively go silent or hide, or we also call that latent infection. But the virus is still present, and at any time the virus can come out of that cell, or the cell might get activated and release viral particles. But in a person on treatment, the virus can't go on to infect new cells. So while the person's on treatment, everything stays under control. Very low levels of virus that aren't measured by the tests that we use in the clinic. And these very persistent, long-lived, latently infected cells. Now, the latently infected cells are actually really rare. Only about 1 in a million, in a person on treatment, only about 1 in a million cells carries this latent virus, but it persists for the lifespan of the individual. So a lot of work is aimed at trying to, first of all, find ways to pick this latent infected cell up. It's very hard to detect it, and ways we can eliminate it. The other place that the virus hides, and again, many people in the Burnet working on this as well, including our colleague Melissa Churchill, who's in the room, is how the virus can also hide in the brain, in the gut, in the genitourinary tract, and in lymphoid tissue. So that's the problem. The problem are these long-lived, latently infected cells, the virus hiding in certain tissues at very low levels. I'm going to just talk a little bit about a few projects my own group is doing in understanding and trying to eliminate latency. Because if we could eliminate those latently infected cells, perhaps we could find a cure, perhaps functional cure or sterilising cure. So first of all, this is a big group effort. Unfortunately, this is an old photo, so I don't have everyone in my group pictured here, but there's about 25 people in the group working with many collaborators, both within the Burnet, within Australia and internationally, on this effort to understand latency and eliminate it. And I'm going to show you a few projects that are led by different members of my group. And one of the key projects is led by Paul Cameron, who's also in the audience, and Suha Sayer. And what Paul and Suha discovered several years ago is how to create a latently infected cell in a test tube. And that has actually been quite difficult. We can very easily grow HIV when it's replicating, but not to create a latently infected cell in the test tube. This is a really valuable resource for us because we can understand a whole lot about how the virus gets in, how we might be able to detect it in people, and how we could perhaps eliminate it. And that's led to a very big collaborative project that's funded by the National Institutes of Health, the major funder of medical research in the US. It's work we do with colleagues in San Francisco and Florida, and with Gabby Curry, Miranda Smith, and Jenny Anderson, who's also in the room. And what we're working on is trying to find new ways to pick up this latently infected cell in patient material. And we're using some of the clues we have from our test tube model of latency and asking, can we use that to pull out latently infected cells from people? And probably one of the more exciting areas, because now this has moved into a clinical trial, is getting rid of latently infected cells by basically waking up the virus. This is a bit counterintuitive to what many of you will understand when we treat for HIV. We use these drugs to lower the virus to extremely low levels. But we think that if we can wake up the virus, this will mean the virus is now visible to the person's immune system or even visible to the drugs, and then the cell will die. And the principle behind this is, if this is a schematic of a cell with its latent HIV, the virus sitting inside the person's DNA, if we could activate that virus, the virus would then start making proteins shown in orange or new viruses, and this would lead to death of the cell, either because this is now visible to the immune system or visible to the drugs. And in the laboratory, we've got a whole lot of different drugs that can do this. The names are not important, but both our group as well as groups around the world have identified many different drugs in the laboratory that can actually do this, activate latent HIV. And several years ago, we became very interested in one group of drugs called HDAC inhibitors. These are drugs used for the treatment of cancer. They're licensed drugs now for the treatment of rare forms of skin cancer. And we thought, you know, this is a very attractive drug to test this idea, whether you can wake up latent HIV in people and do it safely, because we know lots about these drugs already. They've been in lots of people, so we know what their side effect profile is. So very quickly after discovering this in the lab, we were able to set up a clinical trial to test this idea. Can you wake up latent virus in people? Can you do it safely? And perhaps one day eliminate latently infected cells. And we did this clinical trial here at the Alfred. It was the first such trial internationally. It was led at the Alfred by Julian Elliott and a huge laboratory team, but primarily Fiona Whiteman, who's in the audience, who did all the immunology, and Ajantha Solomon, who was responsible for measuring all the different parts of the virus. We worked with the National Association of People Living with HIV to do this clinical trial, and they were that partnership. It's a partnership that really characterises the response to HIV in Australia. Fantastic partnerships between the community and scientists, and we experienced this with NAPWA, who really helped us explain this very complex study with a lot of ethical issues to think about to the community, and we had tremendous support from the community for participation in the trial. We also worked with scientists at Peter Mac, who were experts in these drugs and how they're used in cancer. So this is what the study was. We had 20 HIV positive patients on antiretroviral therapy enroll in the trial. They all had very low levels of virus, very good immune systems, completely healthy. And we asked them to take 14 days of this drug, Verinostat, an HDAC inhibitor. And we measured the amount of virus inside the latently infected cell in blood, before and after they received Verinostat. We have a whole lot of very elegant tests that we can do in the laboratory that can measure these parts of the virus. And what we found was that in 80% of the patients that participated, we could wake up the virus. We could clearly see, measure virus in the cell that wasn't there before. And we also found that the virus didn't go crazy. By waking it up, we saw little bits of virus being released, but it didn't suddenly reactivate the whole cycle again. After 2 weeks of the drug, we didn't see any loss of these latently infected cells. But this is really just the first step, proof of concept that you can wake up the latent virus. And we're now planning other studies with collaborators largely in San Francisco and also within Australia to test this concept again in a slightly different design. And finally, we're very interested in how these drugs work in other places, such as the brain. And we're doing this work with Melissa, an expert on HIV in the brain, and Lachlan Gray and Hao Lu, and looking in the laboratory what these drugs do to different cells, one particular cell called an astrocyte, because we want to also understand what it's going to do to any HIV sitting inside the brain. Now just finally, I want to talk about some work not being done by our own group, but just so you can understand where the science is taking us and what might be possible in who knows how long. And it's another totally different way to think about curing HIV, and this is to make cells or people resistant to HIV. So I started off telling you about people that are CCR5 negative, 1% of the population, really trying to make people's cells CCR5 negative but not giving them a bone marrow transplant. And the way this is being done is with gene therapy. And gene therapy means that we now have the capacity to actually cut out a gene we no longer want. Unfortunately, DNA is not as big as that, so it's a little bit harder than just taking your scissors and chopping out the gene you don't want anymore. But we can do it at a very, very micro level. And what scientists in the US are now doing is they're using these gene scissors. They're literally like scissors to cut out the CCR5, that same protein that I said that the virus needs in order to get inside a cell. And we know that if we remove CCR5 from people's genes, they're going to be okay, because in nature we know that people who naturally don't express CCR5 are fine. They have no defects in their immune system. And if we did that, then the virus would have nowhere to go. So that would be another way to cure HIV. And this is already happening in the US. This is where we're up to at the moment. People with HIV on treatment having very large amounts of blood collected. And when you collect the blood, the immune cells, some of them have CCR5 and some of them don't. And then these cells undergo gene therapy or the gene scissors. You can actually cut out CCR5 and make all those cells CCR5 negative and then put them back into the patient. This has already been done in about 20 or 30 patients. The CCR5 modified cells persist and at low levels currently, but the next step will be to get it at very high levels. And so far, from what we understand, it's been safe and well tolerated. So just finally, when will we have a cure for HIV? I always get asked this question, primarily by journalists, and I always answer it by saying, I'm a scientist, not a fortune teller. I really don't know, and I actually really don't know. I'm very reluctant to put a time frame on it. When HIV first came about, most people and very eminent scientists thought finding a vaccine would take us 5 or 10 years, and 30 years later we don't have a vaccine. And that's because the science has been so complicated. HIV is such a clever virus. We might find a way to attack it one way and it will have another trick up its sleeve. So we have come a long way in understanding why HIV persists, but it still might be a very long way till we have a cure. And I think ultimately the cure that we want is a cure that's going to be available to everyone who needs it. So not a high-tech gene therapy trial where only very wealthy people in one country will be able to access it. We ultimately want a cure that's scalable and accessible to all people. But I do have some optimism that things are going to change a lot in the next 5 to 10 years. And the optimism is based on, first of all, these cases that we know cure is possible. Second of all, there's been a really significant commitment by many funders around the world in placing finding a cure as a top priority on the research agenda, and it's certainly the case in the US, driven by the National Institutes of Health. Our own Medical Research Council have also funded a lot of work in this area, and many, many other organisations are really making a very strong financial commitment. Now, money alone is not going to lead to a cure. We know great science takes a lot more than that. It takes very smart people, it takes collaborations, it takes incentives, but money also will make a very big difference in order to really push and accelerate this field. So finally, Marilyn mentioned that next year we're very excited to be hosting AIDS 2014, or the International AIDS Conference in Melbourne. I'm very privileged to be co-chairing that meeting. It will be the largest medical conference ever held in Australia, but more importantly, this meeting is much, much more than a medical conference. Of the 18 or 20,000 people that we expect, about 5,000 will be clinicians and scientists, and we hope we're going to hear some exciting science in all areas of research at the meeting. But more importantly, the meeting is for communities, it's for politicians, it's for advocates, for discussing issues around human rights and ethics. It's about raising awareness of HIV, and I think we're going to have an amazing opportunity to do that within Australia. The meeting usually takes over a city, so I hope you're going to know well and truly about this in July next year. There's a lot of events planned around the conference that with collaborations with the arts communities, music, dance, etc. There'll be areas within the conference centre that are open to the public that you won't need a registration for, and so I think it will be a very, very exciting venue for all of you who I think have shown a commitment to the Burnet, a commitment to HIV, and I'm sure you'll get to enjoy the conference as well. Thanks very much.